Welcome to our comprehensive guide on biomedical research design. In this video, we'll explore the foundational principles that guide scientific inquiry in the biomedical field. Biomedical research is essential for advancing our understanding of health, disease, and treatment. Whether you're a student, a professional, or simply curious, understanding research design is crucial for interpreting scientific findings and contributing to the field. We'll start by defining what biomedical research is, then move on to the key elements that make up a solid research design. By the end, you'll have a clear roadmap for planning, conducting, and evaluating biomedical studies. Let's begin our journey into the world of scientific discovery. Biomedical research refers to the scientific investigation of biological processes and diseases, aiming to improve health outcomes. It encompasses a wide range of studies, from basic laboratory experiments to clinical trials involving human participants. The ultimate goal is to generate knowledge that can be translated into better diagnostics, treatments, and preventive strategies. Biomedical research is multidisciplinary, drawing on biology, chemistry, medicine, and even engineering. Understanding the scope of biomedical research helps us appreciate the complexity and importance of designing robust studies that can withstand scientific scrutiny. Well-structured research design is the backbone of any successful biomedical study. It provides a clear plan for collecting, analyzing, and interpreting data. Without a solid design, studies may yield unreliable or biased results, wasting valuable resources and potentially leading to incorrect conclusions. Research design ensures that the study addresses the research question effectively, controls for confounding variables, and produces results that can be replicated and trusted. In biomedical research, where findings can impact patient care and public health, the stakes are especially high. Biomedical research can be broadly categorized into basic, translational, and clinical research. Basic research focuses on understanding fundamental biological mechanisms, often at the molecular or cellular level. Translational research bridges the gap between laboratory discoveries and clinical applications, aiming to move promising findings into real-world practice. Clinical research involves studies with human participants, such as clinical trials, to evaluate new treatments or interventions. Each type plays a unique role in the continuum of biomedical discovery, and understanding their differences is key to designing appropriate studies. Every successful biomedical study starts with a well-defined research question. This question guides the entire research process, from selecting methods to interpreting results. A good research question is specific, measurable, and relevant to current scientific or clinical challenges. It should address a gap in knowledge and be feasible to answer with available resources. Taking the time to refine your research question ensures that your study remains focused and impactful, setting the stage for meaningful scientific contributions. Once the research question is set, the next step is to formulate a hypothesis. A hypothesis is a testable statement predicting the relationship between variables. In biomedical research, hypotheses often propose how a particular intervention or exposure affects health outcomes. Clear, well-constructed hypotheses help researchers design experiments, choose appropriate measurements, and interpret findings. They also provide a framework for statistical analysis, making it easier to draw meaningful conclusions from the data. Understanding variables is fundamental to research design. Variables are characteristics or factors that can change or be manipulated in a study. In biomedical research, we often distinguish between independent variables, what you change, dependent variables, what you measure, and confounding variables, other factors that might influence results. Clearly defining and controlling variables helps ensure that your study accurately tests the hypothesis and produces valid, interpretable results. Choosing the right study population is crucial for biomedical research. A population includes all individuals who meet the criteria for participation in your study. Defining inclusion and exclusion criteria helps ensure that the study results are applicable to the intended group. Careful selection minimizes bias and increases the generalizability of findings. Whether working with cell lines, animal models, or human participants, population selection is a key step in research design. Sampling refers to the process of selecting a subset of individuals from the study population to participate in the research. Common sampling methods include random sampling, stratified sampling, and convenience sampling. The choice of sampling method affects the representativeness of your sample and the validity of your conclusions. Random sampling is often preferred because it reduces selection bias, but practical considerations sometimes require alternative approaches. Ethics are at the heart of biomedical research. Researchers must ensure that studies are conducted with respect for participants' rights, safety, and privacy. This includes obtaining informed consent, protecting confidential information, and minimizing risks. Ethical review boards or institutional review boards, IRBs, evaluate research proposals to ensure compliance with ethical standards. Adhering to ethical principles not only protects participants, but also enhances the credibility and acceptance of your research. Biomedical research designs can be broadly classified as experimental or observational. Experimental studies, such as randomized controlled trials, involve actively intervening and observing the effects. Observational studies like cohort or case control studies involve monitoring participants without intervention. Each approach has strengths and limitations, and the choice depends on the research question, ethical considerations, and available resources. Randomized controlled trials are considered the gold standard in clinical research. In RCTs, participants are randomly assigned to intervention or control groups, minimizing bias and confounding. This design allows researchers to establish causal relationships between interventions and outcomes. RCTs are widely used to test new drugs, therapies, or preventive measures, and their rigorous methodology makes their findings highly reliable. Cohort studies are a type of observational research where a group of people sharing a common characteristic is followed over time. Researchers observe outcomes, such as the development of disease, in relation to exposures or risk factors. Cohort studies are valuable for studying the natural history of diseases and identifying potential causes, especially when experimental studies are not feasible. Case control studies compare individuals with a specific condition cases to those without it controls. Researchers look backward to identify exposures or risk factors that may have contributed to the condition. This design is efficient for studying rare diseases or outcomes and can provide valuable insights, though it is more susceptible to bias than some other designs. Cross-sectional studies assess a population at a single point in time. They are useful for measuring the prevalence of diseases or health-related factors and for generating hypotheses. While they cannot establish causality, cross-sectional studies are relatively quick and cost-effective, making them popular in public health research. Longitudinal studies involve repeated observations of the same subjects over an extended period. This design allows researchers to track changes, identify trends, and infer temporal relationships between exposures and outcomes. Longitudinal studies are powerful tools for understanding disease progression and the long-term effects of interventions. Blinding and randomization are key strategies for reducing bias in biomedical research. Randomization ensures that participants are assigned to groups by chance, while blinding prevents participants and researchers from knowing group assignments. These techniques help ensure that observed effects are due to the intervention, not other factors, and increase the credibility of study findings. Control groups are essential for comparing the effects of an intervention against a baseline. In biomedical research, control groups may receive a placebo, standard treatment, or no intervention. Including a control group helps isolate the effect of the experimental treatment and strengthens the validity of the study's conclusions. Determining the appropriate sample size is a critical step in research design. Too small a sample may lack the power to detect meaningful effects, while too large a sample can waste resources. Sample size calculations consider factors like expected effect size, variability, and desired statistical significance. Proper planning ensures that your study is both efficient and scientifically robust. Data collection is the process of gathering information relevant to your research question. Methods can include surveys, interviews, laboratory tests, medical records, or wearable devices. Choosing the right data collection method depends on the study design, population, and variables of interest. Accurate and consistent data collection is vital for producing reliable results. Measurement tools are instruments or procedures used to assess variables in a study. In biomedical research, these can range from laboratory assays and imaging techniques to questionnaires and physical exams. Selecting valid and reliable measurement tools is essential for ensuring that the data accurately reflect the variables being studied. Effective data management involves organizing, storing, and protecting research data throughout the study. This includes creating data entry protocols, maintaining secure databases, and ensuring data integrity. Good data management practices facilitate accurate analysis, reproducibility, and compliance with ethical and legal requirements. Statistical analysis is used to interpret research data and determine whether observed effects are significant. Common techniques include descriptive statistics, hypothesis testing, regression analysis, and more. Choosing the right statistical methods depends on the study design and data type. Proper analysis helps draw valid conclusions and supports evidence-based decision-making. Bias refers to systematic errors that can distort study results. Common types include selection bias, measurement bias, and publication bias. Recognizing and minimizing bias is essential for producing valid and trustworthy findings. Strategies include randomization, blinding, and careful study design. Confounding factors are variables that can influence both the independent and dependent variables, potentially distorting the true relationship between them. Identifying and controlling for confounders is a key part of research design. Techniques include randomization, matching, and statistical adjustment. Validity refers to how well a study measures what it intends to measure, while reliability is about the consistency of results. High validity and reliability are essential for credible research. Researchers use standardized protocols, validated tools, and repeated measurements to enhance these qualities. Pilot studies are small-scale preliminary studies conducted before the main research. They help test the feasibility of the study design, identify potential problems, and refine methods. Conducting a pilot study can save time and resources by ensuring that the main study is well-planned and likely to succeed. Interpreting data involves making sense of the results in the context of the research question and existing knowledge. Researchers consider statistical significance, clinical relevance, and potential limitations. Clear interpretation helps translate findings into actionable insights for science and medicine. Accurate and transparent reporting of research findings is essential for scientific progress. Researchers should provide detailed descriptions of methods, results, and limitations. Following reporting guidelines such as Consort for clinical trials helps ensure that studies can be evaluated, replicated, and built upon by others. Peer review is a critical step in the scientific publication process. Independent experts evaluate the study's design, methods, and conclusions to ensure quality and validity. Constructive feedback from peer reviewers helps improve the research and maintains the integrity of the scientific literature. Systematic reviews and meta-analyses synthesize evidence from multiple studies to provide a comprehensive understanding of a research question. Systematic reviews follow structured methods to identify, evaluate, and summarize relevant studies, while meta-analyses use statistical techniques to combine results. These approaches are invaluable for evidence-based medicine. Translational research aims to move discoveries from the laboratory to clinical practice. It bridges the gap between basic science and patient care, ensuring that promising findings lead to real-world health improvements. This process involves collaboration among scientists, clinicians, and industry partners. Implementation science studies how to best integrate research findings into routine healthcare practice. It addresses barriers to adoption, evaluates strategies for change, and measures the impact on patient outcomes. Effective implementation ensures that scientific advances benefit as many people as possible. Reproducibility means that other researchers can repeat a study and obtain similar results. It is a cornerstone of scientific credibility. Ensuring reproducibility involves transparent reporting, sharing data and methods, and adhering to rigorous standards throughout the research process. Open science promotes transparency, collaboration, and accessibility in research. Practices include sharing data, publishing open-access articles, and pre-registering study protocols. Open science helps accelerate discovery, reduce duplication, and foster trust in scientific findings. All research designs have limitations such as sample size constraints, measurement errors, or unmeasured confounders. Recognizing and addressing these challenges is essential for interpreting results accurately and planning future studies. Honest discussion of limitations enhances the credibility of your research. Securing adequate funding and resources is a practical aspect of research design. Funding supports personnel, equipment, data collection, and analysis. Researchers must develop detailed budgets and seek grants from government agencies, foundations, or industry partners to bring their studies to life. Collaboration brings together diverse expertise and perspectives, enhancing the quality and impact of biomedical research. Multidisciplinary teams can tackle complex problems more effectively, share resources, and foster innovation. Building strong collaborations is key to advancing scientific discovery. Disseminating research findings ensures that new knowledge reaches the scientific community, healthcare providers, and the public. Methods include publishing in journals, presenting at conferences, and using digital media. Effective dissemination maximizes the impact of your research and promotes evidence-based practice. Research findings can inform health policy and guide decision-making at local, national, or global levels. Engaging with policymakers and stakeholders helps ensure that evidence is used to improve health systems and outcomes. Translating research into policy is a vital step in maximizing the societal benefits of biomedical research. Continuous quality improvement involves regularly evaluating and refining research processes to enhance efficiency and outcomes. This may include updating protocols, adopting new technologies, or incorporating feedback from previous studies. Ongoing improvement ensures that biomedical research remains relevant and impactful. Emerging technologies such as artificial intelligence, genomics, and wearable devices are transforming biomedical research. These tools enable more precise measurements, larger datasets, and novel study designs. Staying informed about technological advances can open new possibilities for research and discovery. Global health research addresses health challenges that cross national boundaries. It involves collaboration among countries and disciplines to tackle issues like infectious diseases, health disparities, and access to care. Designing studies for global health requires cultural sensitivity, adaptability, and a focus on equity. The field of biomedical research design is constantly evolving. Future directions include greater use of personalized medicine, adaptive trial designs, and real-world data. Embracing innovation and flexibility will help researchers address emerging health challenges and continue making meaningful contributions to science and society. Summary: mastering biomedical research design is essential for advancing health and scientific knowledge. From defining research questions to implementing findings in practice, each step requires careful planning, ethical consideration, and a commitment to quality. By understanding and applying the principles covered in this video, you'll be well equipped to contribute to impactful biomedical research and help shape the future of healthcare.